Let me introduce myself. I am Dr. Rani K. Pillai, Assistant Professor, Department of Physics, NSS College, Nilamir. Here I present a paper, Anti-Cancerous Activity of Green Synthesized Alpha Manganese Oxide on A549 Human Lung Cancer Cell. Green synthesis of nanoparticles aims at minimizing generated waste and implementing sustainable process. Green synthesis of nanoparticle represents an advance over the method because it is simple, cost-effective, and relatively reproducible, and often results in more stable materials. As an introduction, the treatment of cancer involves different therapies based on alkylating agents, anti-metabolites, biological agents, etc. The phytochemical properties of the nanoparticles, their anti-cancer activity, are either related to intrinsic features or depend on the activities based on the application of external stimuli. In the materials and methods, the fresh leaves, the nanoparticles is synthesized from the tulsi leaves, commonly called tulsi leaves, Ocimum tinifolium. These leaves was collected and thoroughly washed with the distilled water. The leaves are chopped into fine pieces and boiled into 200 milliliters of distilled water for 10 minutes. The extract was filtered and stored in 1 degree centigrade and then used for nanoparticle synthesis. Here we can see that the leaf extract was taken in a 500 milliliter of flask and placed in a magnetic stirrer, and 1 millimolar solution of the potassium permanganate is added. The plant extract is mixed with the 1:1 ratio of 1 millimolar potassium permanganate solution using a burette and stirred for 6 hours. The formation of the red color was observed. The solution is stored in the room temperature for 24 hours and for the complete settlement of nanoparticles. After 24 hours, discard this supernatant. Add 1 milliliter of distilled water to solution and washed. This precipitate is collected by using Watsman filter paper and dry in a batch glass and store as the nanoparticles. These nanoparticles is used for undergo annealing. These annealed samples of manganese oxide was characterized using XRD, SEM, TEM, and FTIR. The notable line broadening of the diffraction peaks is an indication that synthesized materials are in a nanometer range. The average grain size of these particles has been determined by the Scherrer formula. The surface morphology of the powdered sample was obtained by the SEM, the scanning electron microscope. The SEM image is a well-defined thread-like morphology for the prepared of alpha manganese oxide particles. The TEM study suggested that the rod-like morphologies for the obtained alpha manganese oxide particles. In FTIR spectrum, a prominent peak is observed in 3,500 range, and it is due to the OH vibration present in the material and may be occluded the water molecules, and the observed peak in the range of 500 to 550 nano range clearly indicate the presence of the manganese oxide formation. No other peaks are observed in the spectra, even if the leaf extracts contains so many functionalities indicate the observed material is purely nanomaterial. According to the anti-cancerous activity, the plant-derived drugs are desired for anti-cancerous treatment as they are natural and readily available. They can be readily or orally as part of the patient's food. Also, being naturally derived compounds from the plants, they are more generated, more tolerated, and non-toxic to normal human cells. In this study, the anti-cancerous analysis done in A549 human cancer cell using MTT-SA method shows moderately active results shows in the tabular column. As a conclusion, the alpha manganese oxide nanoparticles is prepared from tulsi leaves extract. The TEM study suggested the rod-like morphologies are the obtained alpha manganese oxide nanoparticles. The percentage of validity shows the material is moderately active towards the A549 human lung cancer cell. This is the reference, and I acknowledge the characterization for the stick poaching for the characterization techniques. Thank you.